Hello, and welcome to the next edition of the Science Education Series. Today, I am with Dr. Sreevidya Meesala, who is our Senior Fermentation Scientist here at BIO-CAT Microbials. Thank you for being with us today.
Thanks, Caroline. Thanks for the opportunity.
Absolutely. Well, I'll just go ahead and jump right in. So the first thing I'd love to do is discuss your background. So what was your draw to the world of fermentation?
My journey into the world of fermentation began with a deep fascination for how microorganisms can drive large-scale biochemical processes. During my academic years, I was particularly drawn to the complexity and potential of microbial systems in producing everything from pharmaceuticals to biofuels. This interest solidified as I gained hands-on experience in the lab, where I saw first-hand impact of optimizing fermentation processes. Over the years, I have had the opportunity to work on diverse fermentation projects, both upstream and downstream processes. So in my previous work, I worked with bacteria, actinomycetes, and fungal strains. What continues to excite me about fermentation is its versatility and the constant evolution of the field. The ability to harness and manipulate the biological processes to solve real-world problems is both challenging and rewarding, and it's what keeps me motivated in my work.
Next, could you walk us through a typical fermentation process here at BIO-CAT Microbials?
Sure. At BIO-CAT Microbials R&D, we develop and optimize various aspects of the fermentation process at benchtop and pilot scale to produce bacillus spores. These spores are then produced in our production facility using spray drying with rigorous quality checks conducted at every step of the process. We have diverse collection of over 2,000 bacillus strains which we supply to customers for various applications, including probiotics for animal feed, human consumption, and also agricultural use.
Next, what variables can impact how long it would typically take? And what are some of the key factors that you consider when working with microbials in this way?
The primary variables that impact fermentation process include temperature, pH, and the carbon-nitrogen ratio, the percentage of dissolved oxygen in the fermentation medium. Given that our products are stable spores, optimizing bacillus sporulation is a critical focus in our bioprocess development. For each strain, we not only determine the optimal conditions for growth, but also the best conditions for sporulation. The art and science of optimization process lie in finding the right balance between providing enough nutrients for the growth and at the same time withholding the nutrients to trigger sporulation. Achieving this balance can be challenging as it varies depending on the strain. Some strains are particularly finicky, but we are always up to the challenge of optimizing them for the best possible outcomes.
So lastly, how is the fermentation process different for spore formers versus non-spore forming bacteria?
Yeah, it's a good question. The primary difference is the unique life cycle of spore forming bacteria. For spore formers, a key challenge is managing the transition from vegetative state to spore formation. Conditions needs to be optimized to trigger and enhance sporulation, which often involves nutrient limitation or some stress factors. For non-spore formers, optimizing the conditions for growth, product formation, and maintaining culture viability is important without the added complexity of managing a sporulation cycle. In summary, the difference lies in the need to carefully manage the life cycle stages of spore forming bacteria, which adds an additional layer of complexity to the fermentation process compared to non-spore formers.
Great. Thank you so much for chatting with us today. We've really enjoyed the opportunity to hear your perspective.
Thank you so much.